Good evening. My name is Enrico Bernardo, and I'm the Vice Chair for the Chancellor's Associates, and I'm also an alumnus for UC San Diego. It's great to see all of you here, another full house, so thank you all for being here. For those of you first time attending in here, welcome. And these events, we host about 2 or 3 of these a year, and we really want to highlight some of the events and the work that the campus is doing, and this one as well. Super excited to be here. We hope you find this event tonight enjoyable. So just a little bit about the Chancellor's Associates. Since 2013, our community has worked in partnership with Dr. Khosla to award more than 2,500 full ride scholarships to scholars. So 2,500, and these go to high-achieving California students whose families lack the financial resources to send them to college. So what's special about this program, though, it's a wraparound service. Not only is it the scholarship and the funds, but we also provide services to ensure that students, our scholars, thrive and receive assistance when needed and remain fully engaged both in academic and campus experiences. So it's really both the finance as well as the support that comes around. So I wanted to highlight who is in this room tonight. We have CASP scholars here tonight, alumni, staff, we have faculty emeriti in here, leaders from around campus, and a special thanks to the Chancellor's Associates members and supporters. All of you have helped grow this program and help provide this opportunity for these young scholars. So thank you. Because of your passion, whether that's through your generosity or just being here and giving these students their chance to shine, we really want to thank you for your time tonight. So appreciate that one. So at the end of this event, if you are interested in being part of the Chancellor's Associates, please come see any of us. We have some associates around in here. We'd love to talk to you and see some opportunities in there. So tonight we have Dr. Dean Christine Hrycyna speaking to us, but before she speaks to us, I'm really excited to introduce a young scholar, Isaac Castillo. He is a major in biochemistry, minor in psychology, 4th year student, and he's going to speak to us about his experiences as a Chancellor's Associate. So Isaac, please.
Thank you, Enrico. So, hi everyone. My name is Isaac Castillo, as you probably already heard. I am a 4th year biochemistry major with a psychology minor over at Muir College, with the goals to become a practicing physician one day. Growing up, I was told by my parents, who were not able to gain a college education on their own, how much of a great privilege it is to learn, and that when one works hard, there will always be a door that will open. Back in 2021, when the world was filled with uncertainty as the pandemic continued to scar the world, I still remember that day that I received my college acceptance to UCSD, and shortly after realizing I had been given the CAS scholarship. I remember that day being one of joy and bright colors as I rushed to my parents to tell them the great news. I realized right then and there I had been given the privilege of a lifetime. Although through all that excitement, moving away from home was perhaps the most hardest part of my journey here at UCSD, practically because we were all in lockdown for more than a year. However, through the CAS program, I was able to receive amazing guidance from the staff and my mentor that all helped me during my transition here. And it was made clear to me right then and there that regardless of what the circumstance would be, there would always be a spot here on campus with a phenomenal group of people that would always believe in me and my journey. During my time here at UCSD, I've had the great opportunity to learn new things and take a wide scope of classes, from learning how scientists carry out drug design, to learning the mechanism and effects drugs have, to this day learning how scientists are able to carry out genome editing to potentially serve a way to cure many diseases. All of this knowledge that I will carry with me as I work towards my ultimate goal of medicine. I have also been given the great opportunity to perform research and use novel tools to better understand human mechanisms to design interventions for neurological disorders. And from this, I've been able to understand just how complex clinical research can be, while also gaining hands-on experience. In addition, being a part of a protein biochemistry research fellowship and being able to volunteer here at our UCSD Health Hospital has further nourished my passion for not just medicine, but also science. Opportunities such as these are a very small fraction in comparison to the overwhelming amount of opportunities and support available here to UCSD students to help them reach not just their goals, but also their full potential. Now, ever since last year, I made it a goal of mine to advance the CAS mission and give back to the program that has given so much to me. So I started off as an Oasis Learning Community Facilitator, in which I work with amazing CAS staff members that are over there in the back of the room, in which we lead sessions geared towards first-year students. In these sessions, we teach them about the many resources that are available to them, while also fostering a sense of community and belonging. This year, I've also been given an opportunity to become a CAS peer leader, in which I'm able to work closely with 16 first-year CAS scholars. And through this position, I am always amazed to hear about the different backgrounds that my mentees come from and their future aspirations, and being able to see firsthand just how CAS is a bridge to reaching them. Without mentorship, I would not be where I am standing today. So as someone who has greatly benefited from mentorship, shout out to my mentor, I can speak with how much of an impact one can have on someone, and how these relationships can take you a long way and can last also a very long time. Therefore, being able to offer holistic support is something that I have always yearned to do. And from this position, I've been able to learn the importance of listening, sharing, empathy, advocacy, and being open to constantly learn, all of which I will carry with me as I embark with what comes next. Now, as I near my graduation this year, I am always beyond thankful for the great opportunity that this program has given me and has continued to give many other students. My next steps will be to study for the MCAT and gain more patient-centered experience as I prepare myself to apply for medical school. Now, I want to say that what you have heard from me right now is, from my experience at UCSD and CASP, is not even the full picture of just how amazing impact this program has on the life of many students like mine. So to all the Chancellor's Associate supporters, staff, and alumni, I would like to thank you for being able to make this one-of-a-kind program a possibility. I can assure you here that your contributions are never left unseen, but rather help pave the way for the leaders and change makers of tomorrow. So thank you. Thank you, Nico.
Thank you, Isaac, for sharing that. More impressive than all your studies and your education is your ability to give back. So we really thank you for that. And we cannot wait to see where you go from here and watch you blossom. So it's now my pleasure to introduce our featured speaker, Dean Christine Hrycyna. Dr. Hrycyna is currently the Dean of the School of Physical Sciences and the Chancellor's Associate's Chair in Chemistry and Biochemistry for UC San Diego. I'm going to read this carefully. This is amazing. Dean Hrycyna's research focuses on the biochemical basis of aging disorders and cancer and the development of new treatments for brain, pancreatic, and lung cancers. She currently serves on the editorial board of the Journal of Biological Chemistry and has served as a standing member of the peer review committee on cancer drug development of the American Cancer Society. Please join me in welcoming Dr. Hrycyna.
Thank you. Oh, did you turn this one off? Oh, I'll use this one for a few minutes. Well, Isaac, that's a hard act to follow. Man, how impressive was Isaac? Another round of applause, please, for Isaac. Unbelievable. It reminds me a bit of my own story, which I hadn't realized until you started talking. I grew up in Connecticut, a child of immigrant parents that fled World War II from Ukraine when they were children. So we ended up in Hartford, right outside of Hartford, Connecticut, and didn't have huge resources to go to college. And without that support, I went to Middlebury College in Vermont, a small liberal arts college, but the story couldn't be more similar. I would not be here today without that support. So I'm really glad that you were able to get that support. I also wanted to go to medical school. And then I shadowed a physician, and then I realized you actually have to like touch people and their fluids, right? And that's when I pivoted back to my research lab and never looked back. But I will want to say, MD, PhD, right? That's what I'm going to say. So what a delight to be here. What an incredible program this Chancellor's Associates is. I'm really excited to learn more and more about it. I'm new to UCSD. I started as dean a year and a half ago, and we just moved here a year ago, so last January. I was at Purdue University for 25 years. So I was bleeding black and gold for a long time, and I was like, what's going to get me out of there, right? Well, my husband said, I don't want to die in Lafayette. And I'm like, fair enough. He had been there much longer. He got all his engineering degrees at Purdue. I was like, okay, that's one good reason. Another reason was the talent here. The talent in this university is beyond measure. And from the students to the faculty, I couldn't be more thrilled to be here. And even in these difficult times we find ourselves in, I see the camaraderie building, and that's been very exciting to see. Okay, so I'm going to tell you what I did. All this work was done at Purdue, and where I obviously spent all my life. I was at NIH before that as a postdoctoral fellow, and that's where some of this work started. And I do have some UC roots. I was a graduate student at UCLA because it was really cold in Vermont, and I was like, I'm going to go to UCLA. That sounds great. All right, so what I'm going to tell you about today is work we're doing on getting things across the blood-brain barrier. But first, I'll tell you what the blood-brain barrier is. That would probably be helpful. So the blood-brain barrier, think of it as, a security system for your brain. You don't want every molecule that you're ingesting or in breathing in or whatever entering your brain. That would be incredibly bad. So you have this surveillance system around. But it's not a thing around it. It's within the capillary network within your brain. Now, I don't know exactly how they figured this out, but there's about 375 miles of capillary vessels within your brain, okay? And again, that tightly controls the passage of nutrients and restricts the passage of potentially harmful compounds. We call them xenobiotics, because it sounds fancy, and such as neurotoxic agents. But things you wouldn't even consider that are neurotoxic, if they can enter your brain, become neurotoxic. And the only reason they're not is because they can't get in because of the blood-brain barrier. And so what the blood-brain barrier really is are the cells that line these capillaries. And their job is basically to tightly control what comes in and what goes out of your brain. And there's no leakage at all, right? It's an incredible system. The question is, what if you want to get something in, like a therapeutic, like an antidepressant, like an anti-psychotic agent to treat any kind of brain disease? What do you do? That's where the question lies. So there's a bunch of things you can do, from invasive techniques that are sort of basically disrupt the blood-brain barrier for a period of time and hope it goes back together, okay, using ultrasound or chemicals or osmotic pressure, things like that. You can also do other things, and worrying about modifying the drugs, make them more transparent to the blood-brain barrier. So if something's very hydrophobic, so it has lots of lipid groups on it, or very sticky things that are like oil and water, the oil part will get in because it can just diffuse across the cellular membranes. We happen to be looking at, there's a lot of transporters, little pumps at the blood-brain barrier within these capillaries that pump things out before they can get into the brain. Right? Those are the molecules that we are targeting. So if we look more closely at this close-up of the capillaries, you can see here the capillaries, and this is the brain tissue around it. So the only way things can go in and out of your brain are through these capillary walls. Now, if we look head-on down a capillary, so looking down there, what you'll see is here's the blood in the center of it. Here's the cells that are forming that blood-brain barrier. And in this space here are a bunch of transporter proteins, little molecular pumps that surveil. Their job is a surveillance role. They're surveilling this for anything that they think shouldn't be in the brain. So here's sort of a flattened image of it. Here's the brain side. Here's the blood side. The drugs will come here. Most of them are lipophilic, so they'll dissolve within the membrane of each of these cells. But here's our friend P-glycoprotein. What's short for that is PGP. And it sees the drug and it pumps it right back out. Okay? And so you have a lot of different kinds of pumps at the blood-brain barrier. But the one that we focus on most is this P-glycoprotein because it accounts for probably 90% of all the transport of xenobiotics out of the bloodstream, keeping them out of the brain. Okay, so it's a ubiquitous kind of system. It's not just in the blood-brain barrier. So P-glycoprotein, where is it localized? Well, it's protecting all of the sanctuary sites in your body. The sanctuary sites being the brain capillaries at the blood-brain barrier, precluding access of anti-cancer, anti-HIV, antipsychotic, antidepressants, anything to get into the brain. It's present on the surface of cancer cells. So cancer cells, when you get multi-drug resistance, some of it is due to the overexpression of these pumps, and it keeps the cancer drugs out of the cells. It kills your normal cells, keeps these alive. So if you ever hear about drug resistance in cancer therapies, this is one of the ways that can happen. It's also present in the transporting epithelial cells of your small intestine and your colon. So anything you eat that your body thinks is a toxin will not get into your bloodstream. It'll get pumped back out into your intestines and get excreted. Okay? So that's another one of these sanctuary sites. But this is another thing, and it limits the bioavailability of any drug you take. So you take a pill, and one of the things you have to tell the FDA is how much can it get absorbed into your body. A lot of it is not absorbed because of the action of these transporter pumps. It's also present in placenta to protect the growing fetus. It's present in testes, it's present in liver, and it's present in kidneys. So it has this surveillance role all over your body in order to keep these sanctuary sites safe. So what happens if you knock it out? Okay, in the early 90s, a group of scientists in the Netherlands did these experiments where we knocked out the gene that encodes for P-glycoprotein called MDR1 because it was for multi-drug resistance. So what happens? The mice were perfectly fine. They knocked this gene out. Nowhere on their bodies had any of this protein. They were perfectly fine until they had a toxic insult. And that toxic insult was ivermectin. Ivermectin is used to clean their cages. And it's also HeartGuard, if any of you have dogs. It's the same compound. So what happened was, this is my old person's death, right? It's like, I don't know what to do, so I just put a big X through it, right? They died. They used to be upside down, but I don't know why they turned. So I turned them back over, but anyway, they're dead. So just because they were treated with this, they died. And because it was neurotoxic. So they would convulsions and die. The other compound that, when they started screening the compounds, loperamide, which is Imodium, right? You think of that as just an anti-diarrheal. If you didn't have any MDR, any PGP, you would go into convulsions and have neurotoxicity. So that's how exquisitely controlled these pumps are. So what did we learn from these studies of these knockout mice, and how can we relate that to other mammals? So the mice were fully viable and fertile this is the key, under controlled lab conditions. Toxic insults will cause problems. They're very sensitive to these toxic xenobiotics, especially neurotoxic ones. And pharmacokinetics, which is basically just tells you where molecules end up in your body when you take them, such as these vinca alkaloids, which are used in cancer treatments, fexofenadine, which is Allegra, ivermectin. You got increased gut absorption, decreased kidney and liver excretion, all the sites where you'd expect this to be functioning. Now, this takes us to, so now you're like, all right, Chris, you're now going to turn this off when you try and inhibit this pump so you can get more drugs into the brain? Am I going to die, right? It's a valid question. So, but collies, turns out, 25% of collies in the United States are homozygous for this deletion, like the mice, turns out. Only 1 in 5 collies are homozygous for the normal form. So if you give these dogs ivermectin, they will have neurotoxicity and die. So if you ever have a collie, make sure they get tested for this gene mutation to see if they're positive for MDR, because then you can give them HeartGuard. So you can exist normally and function if you're a collie, but we're assuming that you can do this with a human under controlled conditions. So ABC transporters. So P-glycoprotein is a member of this big family of transporters, about 50 different kinds in the human body. And they sit in the membrane here, so this is the cell membrane. And they have these 2 parts that are embedded in the membrane, and then these 2 parts that are inside the cell. And these use the energy when you break apart adenosine triphosphate, ATP. When you break that, it causes transport of the molecules out of the membrane and back into the bloodstream. So this was the 1990s diagram of what this protein looked like. It really hasn't changed much, even though we have the high-resolution three-dimensional structure. So this was hypothesized by my postdoc advisor. And the way people think about it is it's a hydrophobic vacuum cleaner. Think of it that way. It's sucking things out of the membrane and getting them out of the cell. So before they can get into the cell to do their toxic activity, this surveillance pump is sitting there at the membrane. You're binding it into the protein, and it's using this energy of hydrolysis to spit that compound out. Now, the one thing that was really intriguing to us when we started this project was that they were able to find at least two distinct parts on the protein, these drug binding sites. So where the drug gets recognized and is bound so that it can get pumped out. And when we look at real structures, this one actually is crystallography, X-ray crystallography, but there's some high-resolution cryo-electron microscopy data as well. There's very clear evidence where the substrates are binding into this protein. So we thought, okay, if you have one drug can bind here and get pumped out, what if we block both sites at the same time? Would that get pumped out? Or can we form something that will stop the pump from working? So that was the basis of where we started these experiments. So here's again this picture. One thing I should tell you is that this protein is incredibly promiscuous. It can recognize literally hundreds of different compounds, and that's why it's so effective at being a cleaner of the membranes before they can get into the brain. So I don't know if I know lots of people that take many of these different compounds. So some of them are anti-cancer drugs like Taxol, Gleevec, colchicine, vinblastine, doxorubicin, and we're getting down here into quetiapine, paliperidone. These are antipsychotic agents. But this is just a very small list. So one of the sort of existential questions when it comes to looking at this protein is, how can one protein recognize so many different compounds? And it's still under investigation. But part of it has to do with having these multiple binding domains. So our idea was, how can we reversibly, because you want to be able to turn this on and off, reversibly inhibit, so you'd want to end up like the collie and have it completely turned off, right? Reversibly inhibit P-glycoprotein to increase the bioavailability of therapeutics for brain diseases specifically. And the diseases we looked at, you don't usually think of HIV as a brain disease, but there are sanctuaries of HIV that live inside your brain that need to be eradicated, and that's one of the places we've worked. But what I'm going to tell you about today is some of the work we've done with antipsychotic agents that are used to treat schizophrenia, and we've also just recently done a bunch of work looking at how to treat brain tumors more effectively. So again, our hypothesis here was we're going to develop bivalent inhibitors. So we're going to take one molecule that's a substrate, we're going to link it together to try and block the transporter. Okay? So we take the substrate, we dimerize it, so we take two monomers, link them together into one. And here's, again, a picture of one of our inhibitors that we did by cryo-EM here, showing it bound in the binding sites there. So the idea here is these bivalent inhibitors will take advantage of the multiple binding sites. They should be potent inhibitors because of the bivalent nature. They should bind more effectively and not be able to be transported, and have a slower ability to come off the transporter because they have more energy of binding associated with the protein. So the idea is we've put them together with this tether, and this tether we've designed to be reversible. So we can block the transporter with this drug. This will eventually dissociate and regenerate the original drug once it's inside the cell. So I'll tell you a little bit more about that here. So again, we wanted to make these dimerized substrates. Here's just some of the chemistry that we used. It's not really essential to understanding, but so these are amide bonds that are not cleavable once they're inside your body. And then we had ester bonds or ester bonds with disulfide bonds. So disulfide bond is an oxidized form of a thiol, and once this gets inside your cells, this gets reduced, this breaks apart. This will actually come around, rearrange, and re-establish the drug of choice as its original monomer, okay? So we have both irreversible and reversible strategies. This strategy is the most effective, we found, because these ester bonds are too labile to exist in the bloodstream. They break down too fast before they can get to the target. But the disulfide one will only come apart once it's inside cells, because that's where the reducing environment is. Okay, so here's your normal condition, normal state. Here's the blood, here's the brain, here's the cells in between. Normally what you have, if you want a drug to come into the brain, it's just floating around in the bloodstream, comes here, encounters the protein, gets kicked out. So you have to give a lot more of the drug to get inside the brain. You can overwhelm it eventually, but then you have all these side effects from the compounds. So if you've ever heard of, for antipsychotic agents, tardive dyskinesia, you end up taking, the amount you have to take gives side effects where people end up with these movement disorders, okay? So our goal is, so if we can lower the dose, but get a therapeutic amount inside the cell, we'll have a win-win. You'll lose the side effects, but get the therapeutic effect. So we had two approaches. The first approach was the first one we took, is we're just gonna take some molecule that we know isn't toxic, but is a substrate, link it together, and then feed it with the drug of choice that we wanna get into the brain, okay? So that's one approach. This one is a little, not quite as elegant as what we'd like, but this was sort of a proof of principle. And the example I'm gonna show you real quickly is with quinine. The second approach is more of a Trojan horse approach, okay? So what we do is we're gonna link the molecule of interest to itself and co-administer both the dimer and the monomer. The dimer will enter the transporter, will have more affinity than the monomer. It will block the transporter, so the rest of the molecules, because they're lipophilic, can diffuse across the membrane into the cell. Once these are released, this will break down once inside the cell, also generating more of a drug delivery. It'll basically delivering twice the amount of drug, okay? So these dimeric prodrugs are based on the therapeutic agents. They both inhibit at the blood-brain barrier and revert to the monomeric therapy once inside the cell, okay? So it's a twofer kind of thing. And this is the one I'm going to show you a little bit with quetiapine. So quinine, you've all had quinine if you've ever had a gin and tonic, right? Or just tonic. It's a well-known anti-malarial, but it's also a well-known substrate for this protein. So this was a proof of principle. So we use this OH group here to bind two together to make our dimers. And we made them both, some of them that were irreversible linked together, but otherwise we had ones with either that ester or the disulfide plus the ester, so they could get degraded. Now, we do a lot of different assays, and I'm not going to show you all of them. I'm going to show you the ones that are the most visual, okay? So how can we tell if these things are working to inhibit? Well, we have cell lines that we propagate in the lab. This one, they're usually cancer cells because they you can propagate them for forever, basically. And they overexpress the pump at the surface, okay? So here's our cells. You give it a substrate that happens to be fluorescent so you can watch it. So this substrate's called rhodamine. It's a dye, but it happens to be a substrate of this. Luckily, this thing has hundreds and hundreds of substrates. If you incubate the cells both with the dye, with no inhibitor, the cell will stay dark because the pump is pumping everything out. If you put it with the inhibitor, the pumps will stop working and the cells will light up. So you can monitor this either by confocal microscopy or by fluorescence activated cell sorting if you want to quantify. So here's an example. So here's Q2 is just quinine 2. It's not very elegant naming, but 2 quinines put together is Q2. We have different substrates that are fluorescent, so rhodamine, mitoxantrone, prazosin, and doxorubicin. Without any inhibitor, all the cells are dark because the stuff's getting pumped out. But in the presence of our inhibitor, the cells accumulate that. So that's what's proof that we're blocking that protein, because the dyes can accumulate within the cell. So we did this for a library of our different compounds, and we did it in a way that we could quantify it. And if we tried to do the same experiment with just the monomer, we needed almost 80 to 100 times more of that monomer to do the same effect. So we have an IC50 going from about 100 micromolar down to 1. So it's like 100 times better just by putting two together. Okay, so that was super exciting. The other thing we tried to do is, what's another experiment we can do to make sure that the molecule is the drug of choice is in the cell. So here's some cancer cells. Now, for Taxol, which is a cell-killing agent, so these cells here that are not resistant, you can kill them with 3 nanomolar, okay, 10 to the minus 9th. If you have resistant cells that have lots of this pump, you need 9,200 nanomolar, right? That's how effective this pump is at getting rid of that. If we add our molecule of interest at 4 micromolar, we can decrease that 3,000-fold. Okay, so this is how effective we are at inhibiting that pump. We can reverse that Taxol-resistant phenotype 3,000-fold with our molecule. So it's another way to look at it. The last way to look at it is to look at actual capillaries derived from a rat brain. So if you want to look here, here's our normal picture with the brain, the blood. Here's the PGP pumps. So here's some capillaries. So here they are without any drug and or inhibitor. And then you put in the drug, the fluorescent dye, and the inhibitor. Now, without the inhibitor, the capillary should light up because it's pumping it back into the capillary, okay? With the inhibitor, it should go off into the brain, right? Or the buffer, which is the substitute for the brain here. So as you can see, as we add the inhibitor, we're looking for loss of fluorescence now because we're stopping the pump from keeping it within the capillary. So again, with a fluorescent agent, we can see that loss of fluorescence, showing that in actual capillaries, we can show proof of principle of this phenomenon. So that was the first approach. Our second approach is when we co-administer the dimerized agent itself. So what drug can we dimerize itself? And that's where we get into this molecule called quetiapine, or Seroquel is its brand name. It's an atypical antipsychotic, usually primarily treated to treat schizophrenia, bipolar, insomnia, and it has a number of off-label effects. It does have limited brain access, And because of such, you need to give much higher doses to get the therapeutic concentration within the cells. And then, again, the side effects increase. So again, we want to increase therapeutic effectiveness by lowering the side effect toxicity. So we were going to make homodimeric quetiapine inhibitors, and we call them prodrugs, right, at this point, because when it breaks apart, it reveals the actual drug. So here is the molecule itself. It's another OH that we've dimerized via, and this one only has ester bonds at the moment. So these are the homodimers of that. Again, another sort of cartoon, capillary, the brain. We're feeding it. It blocks the pump so that it can't pump out either the monomer. The dimer blocks the transporter. Once other dimers get in, it can break apart within the cell and again deliver that drug. So we made a number of these molecules as well, and we were able to show here again almost 100-fold more potency over the monomer in when we made a dimer, 50 to 80 times better. Okay, so we made these molecules, and they also had extended plasma stability, so they didn't break down until they got to the site of interest. Here's, again, the capillary experiment. Again, we're looking for loss of fluorescence means that it's effective, and this is the data that got quantified. So these are our positive controls, and with our compound, we can bring to a very potent inhibitor, we can show that that's also being very effective. So both dimers inhibit transport down to the control levels. And finally, the last experiment that we did was we did it in an in situ rat model where they actually injected, our collaborators at the NIH, injected the molecules into the neck of the animal. And we can show that this time we used a radioactive compound, but we got more accumulation of that radioactive compound in the presence of our inhibitor. So we were able to block, without our agent, we got a certain amount into the brain. But once we added our inhibitor, we got an increased level of that. So this is sort of our proof in principle in vivo, in situ model for the effectiveness of our compounds. So those are just two examples. We're looking at a lot of different compounds. We've looked at galantamine dimers, which are used to treat Alzheimer's disease, phenytoin dimers for epilepsy, paliperidone, again another, and paliperidone is another antipsychotic, and venlafaxine, which is an antidepressant. So this is really generalizable to almost every molecule we look at because of the substrate promiscuity. So just to conclude, many therapeutics can't cross the blood-brain barrier due to the activity of this pump. We can increase the brain bioavailability of agents used to treat a number of brain disorders. Reversibly, that's what's key, because it'll eventually be washed out because the linkage between the molecules breaks down. Inhibiting the activity at the blood-brain barrier with these substrates. They work by occupying 2 of the drug binding sites. We have other data that show that more conclusively. We had 2 approaches. One, using the dimeric inhibitor together with the treatment molecule, so 2 separate things, and then our Trojan horse approach by making them prodrug inhibitors that will that inhibit but also deliver the drug. We've been able to show it in brain capillaries as well as in situ brain models. And what we're really hopeful for is that it's going to give us promise that we can treat brain disorders without having to increase the dose, and also get molecules across the blood-brain barrier, like Gleevec, for instance, that is very difficult to get into the brain to treat glioblastomas. And glioblastomas is one of the most deadly brain cancers that you can get. The other one we're looking at is metastases of non-small cell lung cancer, which also metastasize to the brain, which is another target we're using with a different drug. So with that, I just want to thank my longtime collaborator, Jean Chmielewski at Purdue, who is an bioorganic chemist. See, Tom Babinski knows her. Longtime collaborator, I've known her for 25, 30 years. All of this work on the blood-brain barrier is done together, and two of our students, David Miller and Chris Campos, who are at the NIH, they do all the mouse experiments. We don't do those ourselves, and funding and all sorts of things. So thank you.
Thank you so much, Dean Hrycyna.
How was that for time? Was that right?
Oh, that was perfect.
Okay, good.
We've actually got some time for a few questions here, so I'm going to be walking around the room with a microphone. If anybody has any questions for Dean Hrycyna, right here. All right, we got our first one.
Lord, it's a fight.
Good. Thanks for the talk. So we have all these two new.
Who are you?
Jonathan Pollicott.
Hi.
I'm in the Chancellor's. Anyway,
Nice. Thanks for coming.
yeah. So we have these two new drugs that are monoclonal antibodies that treat Alzheimer's disease or supposedly treat Alzheimer's disease. They're FDA approved. How do they get into the brain? Those are huge molecules.
Oh, the antibodies?
Yeah.
You know, I haven't really investigated that exactly. And it's a really good question. I don't know. I'm a small molecule girl. I actually don't know how they get it. Does anybody know? No. I could make something up, but I won't do that. Oh. Oh, hi.
Hi, yeah. Fabulous. They're really exciting stuff.
Thank you.
And you have to be super careful to get everything out of the system that could possibly hitch a ride in this process, because you're just lowering the brain's entire defenses when you do this.
You are not wrong.
Yeah. How do you go about that?
Well, I think you just have to do it. You just have to be. It's about thresholds, right? You just have to do it for a short enough amount of time so we can fine tune how quickly that dimer breaks down. So if it breaks down, you know, and you do it in a less toxic setting, right? You don't have other toxic insults at the same time. But tuning the speed at which that molecule breaks down, I think, is what's key there. That's why you don't want it irreversibly bound. No, Paula.
Hi, Chris. When you're making your dimers, you're trying to occupy two sites simultaneously in the target. Do you have any flexibility in the length of the link that you use?
Yeah, so we have a bunch of studies where we looked at the tether length. And it really doesn't matter, because if it's very flexible, it can fold up and fit, it somehow fits in there. It can't be a rigid structure, so there has to be a lot of mobility. But the actual length doesn't really matter if it's flexible enough. Thank you.
Okay, maybe first question or two. Do you get, when you do a KPUU, do you get a PK/PD disconnect where the concentration plasma unbound has no drug, but there's still drug left behind in the brain? And how fast does it clear from the brain?
Yeah, we haven't done those. We haven't gotten to that stage yet to be doing the hardcore pharmacokinetics dynamics.
And a question I have is, because I'm working on covalence right now. If you did covalently modify the transporter, would it just have to turn over and regenerate itself? And would that be enough time, you know, where you might want to do a irreversible, and then you have the time that it takes for the protein to regenerate to be able to give the drugs that you need, and the person would have to.
Right. So there were some early studies where there were antibodies that were, specific to a different formation of the protein, right? So they bound to it to inhibit it, right? That's kind of what you're talking about.
Yeah.
That's a non-covalent interaction.
Yeah.
But the half-life of this protein is 2 or 3 days.
Oh, okay.
So
All right.
it's
You answered my question.
yeah.
Yeah.
That's too long. That answers his question. That's too long to keep the gate open.
Yeah.
Right.
Okay. All right. Thank you.
Hi.
A great presentation. What really caught my attention was the promiscuity of this proteins, right? I mean, this is really fascinating. We have like 25 million and now I think 25 billion molecules, right?
Yes.
And we live in the age of AI, so is it possible to predict using machine learning or AI which molecules are going to, be blocked by PGP and which ones can go through?
Yeah.
And my flip question to that is, can we use that to design molecules that will be brain penetrant?
Yeah, that's a great question. So far, the only characteristics that are common for this protein is that they're highly lipophilic and potentially partially cationic. There's no other, I mean, there's some pharmacophores that people have been trying to isolate, but then there's always an exception. It's an exceptionally promiscuous protein. There must be allosteric binding going on. There must be a lot of functional dynamics that happens right when something binds to have it be recognized. It's a great question, though. It's something we should look at.
We've got time for another question or two. Jonathan right there in the middle row is going to get the mic.
Hi, Jonathan Ash,
Hi.
Chancellor's Associates, Revell, but I was a political science major, so
Okay.
you may have lost me.
I tried.
Appreciate it. So we hear a lot.
You were a
especially-
poli-sci major in Revell?
Yes, it was a very
Wow.
bad decision.
So you had to take like all that math.
I had to take organic chemistry with Tom Bond.
Oh,
and,
gosh.
And then I came home
And
I
cried.
asked my roommates what the heck just happened.
Yeah.
We hear a lot today about vitamins and supplements and all sorts of new things in that area. But a lot of times we hear about issues with absorption. And then you mentioned that earlier in your presentation with PGP. Can the same sort of technology be utilized for increasing the absorption of some of these new products that are on the market?
Oh, absolutely. Yeah, it can apply to any molecule that's recognized by this transporter. Absolutely. And sometimes when, you got to be a little careful with the supplements, right? Because they're not regulated really in any way. And people will say it's natural, right? So is, doxorubicin, the chemotherapy agent, comes from a plant, right? It'll kill you, but it's natural. And you got to worry about drug-drug interactions too. But yeah, no, absolutely.
Do we have any more questions out there? One more. This will be our last question.
It's hot.
Um,
Hi. Oh, it's you again. Hello.
yeah, sorry, yeah, it's just, this is really fascinating.
Hi, well,
so-
thank you.
Does this apply to, creating a cure or therapy for opioid addiction?
Opioid addiction.
Mm-hmm. By creating a block, dimer or something that.
I'll have to chew on that one. Dimerizing opioids. Yeah. Yeah, it might. Oh. To keep the opioids out. Oh, that would be interesting. I'll chew on that. NIH? Yeah, no, of course, absolutely.
Chancellor's Associates Colloquium, where inspiration happens.
Yeah.
Thank you for that. I appreciate
The problem is
that.
you'll keep
There it
everything
is.
else, Yeah, we'll have to think about that. Yeah, right.
All right. Well, we're going to
All right.
welcome Nico onto the stage here. Thank you so
All
much,
right.
Dean Hrycyna.
Yeah.
Thank you so much, Dr. Hrycyna, for that wonderful presentation. And we have a lot to chew about on the opioid topic. Thank you for bringing that up before we end tonight. So just wanted to say thank you for everyone in here for spending the night tonight with us. I know you have busy schedules. Really, really appreciate that one. And I wanted to extend my deepest gratitude to our Chancellor's Associates and for those who are really contributing to build this community. Students like Isaac and others would not be here without this, so really, truly appreciate all that you've done. And with that, if any of you would like to hear more about the program or want to hear information around it, please come talk to me, Danielle. Reach out to any of us. We'll be happy to speak with you on that one. And please join us. We have another one coming up in May. It's going to be at the Birch Aquarium. It's going to be beautiful over there, so please keep a lookout for your emails. With that, thank you again, Dr. Hrycyna. And yeah, I wish you all a good night. Thank you.